Well, good afternoon. We will be presenting the experiment proposal for the quality control analysis of the macronutrients and micronutrients content in general purpose 2020 foliar fertilizer. This is prepared for Chemistry 4015, Instrumental Methods of Analysis for instructor Maria Liz Crespo. We are Group B, composed of Ezequiel Cruz, Juliara Varga, Danili Arroyo, Claudia Mañán, Anel Ríos, Alondra Maldonado, y Genesis Cordero. We will be presenting the proposal's abstract, a brief introduction to the purpose of this experiment proposal, and the problem it is meant to tackle, explain a little the procedure that it will be used for the analysis, what regions and apparatus will be necessary to complete this analysis, and discuss a little the results that have been found. We'll also touch on the proposed budget and the justification for it, the lean canvas, a conclusion on the results, and is used. 2020 fertilizer is among the most popular fertilizers used commercially. This fertilizer is characterized by having a balance of all the essential nutrients that plants need, and it makes it highly useful in large and small plantations, specifically for food production. Imbalance in the concentration of these ions can cause crops to fail and die, which can cost millions of dollars in losses for farmers across the country. The composition of Treside brand 2020 fertilizer was analyzed using ionic chromatography to determine the concentrations of nitrogen, potassium, and phosphorus, as well as using atomic absorption spectroscopy with graphite furnace to determine the concentrations of manganese, copper, and iron. These concentrations were determined to be 4.56, 0.59, 8.13, 36.37, 15.67, and 60.39 parts per million, respectively, after dilution, and fall within the acceptable range for this type of fertilizer.
In order to know or start talking about the experimental section, we must first know what general purpose fertilizer is and what it contains. General purpose fertilizer, otherwise known as 2020 fertilizer, is a type of fertilizer that can be used for both household plants and crops made for human consumption. It contains 20% of nitrogen, 20% of phosphorus, and 20% of potassium that plants need for their growth, development, and fruitage. It also contains micronutrients such as iron, copper, and manganese. It is important to quantify the amount of each nutrient to make sure that plants are growing correctly. If plants have too much of each nutrient, they can suffer chlorosis of their leaves and, well, death of their roots. If they have too little of it, plants may not develop fruitage and they may not develop correct leaves, which are needed for the plants, for the plants to live. As for the experimental section of this, we use two different techniques to quantify the amount of macronutrients and micronutrients in our sample. The amount of macronutrients was analyzed by ionic chromatography, otherwise known as IC, which is a kind of liquid chromatography that separates anions and cations in a sample. The analysts can then use this concentration or these retention times to quantify and determine the ion concentration in the sample. IC is used in the analysis to determine the concentration of nitrogen, phosphorus, and potassium in form of ammonium, nitrite, phosphates, and calcium oxide. Atomic absorption spectroscopy was applied Atomic absorption spectroscopy was used to quantify the amount of micronutrients in the sample. Atomic absorption spectroscopy uses applied characteristic wavelengths of electromagnetic radiation on samples to determine the analyzed concentration. There's different types of atomic absorption spectroscopy, and these include graphite furnace atomic absorption spectroscopy and flame atomic absorption spectroscopy. In this case, the graphite furnace atomic spectroscopy was used to determine the concentration of copper, manganese, and iron in the sample. Determination of macro and nutrients by ionic chromatography was done by a direct calibration method. The calibration curve was built using 5 standards of increasing concentration of nitrogen, potassium, and phosphate. The table with the data for ammonium can be seen on the right. This data was recovered from chromatograms that were analyzed and integrated by hand, measuring their height and width. Here we can see the calibration curves for our sample and the standards. On top we have the calibration curve for ammonium, and we can see in blue where the standards are, and then in orange and gray the quality controls, and yellow has the fertilizer concentration of ammonium. We can also see the nitrate calibration curve on the bottom. It is important to note that while these graphs do have a high level of linearity with a high value of R squared and a high value of F, these do exhibit bias during the analysis by the analyst. In the ammonium X variable residual plot, we can see how there is a trend to go to the negative values and then a sharp increase, while in the X variable residual plots for nitrates, there is a sharp increase first and then a tendency to stick around the 0 value for residuals. The rest of the data sets of the macronutrients exhibit similar results to those found for the regression of ammonium. A summary of the experimental concentrations can be shown below. The percent difference was determined to be a little bit high for our analysis. One possible explanation for this could be that the fertilizer does not contain the concentrations that were specified on the label. This is highly unlikely as fertilizers pass through various quality control checkpoints to assure their concentrations. Another explanation could be incorrect dilutions of the sample, but that is also highly unlikely, as if that were the case, all of the differences or the percent differences would be either above their theoretical concentration or below, but not a mix of values as shown with the ammonium and phosphate that are above their concentrations and the potassium and nitrate which are below. The most likely explanation for this discrepancy could be the manual integration of the peaks, which would explain why ammonium and phosphate concentrations are higher than expected, but potassium and nitrate concentrations are below than expected.
The determination of micronutrients was performed by atomic absorption spectroscopy. A triplicate analysis of each solution was performed using a PE atomic absorption spectrophotometer with graphite furnace. This was done in order to acquire the average peak area to have an accurate measure. A regression analysis and calibration curves were performed using the concentration and average peak areas value. The relative error for copper obtained was 4.45%, as for manganese, 0.65. Here we can see the data used for the quantitative analysis for copper and for manganese, where we can see the concentration of the standards, the QCs, and the unknown. Also, the absorbance and the standard deviation of the absorbance. These calibration curves were built using 5 standards of increasing concentration, where we can see the QC1, QC2, and the unknown. Here are the graph of the copper and the manganese. The results for iron were analyzed using flame atomic absorption spectroscopy, where solutions were analyzed in triplicate using a Perkin-Elmer atomic absorption spectrophotometer. With the concentration and peak areas, a regression analysis and calibration curve were performed. This was done to determine the concentration of quality control standards and unknown, using the linear equation obtained from the calibration curves. The relative error obtained for iron was 16.52. This percent may be due to the sensitivity of the equipment. Here we can see the data used for the quantitative analysis of iron, where we can observe the concentrations, the absorbance, and the standard deviation of the absorbance. This calibration curve was also built using 5 standards of increasing concentration. These were the calculation and formulas used to obtain the results of our experiment.
Like any project, a budget and justified management of it will be proposed. The following breakdown will cover its use and how to use in detail. The stipulated budget will be used as a form of payments to the employees and researchers, purchase of material, equipment, and reagents. In this table, we can see the breakdown of the remuneration to the analysts, researchers, and chemical assistants. In the first instance, the analytic chemist pays $28 per hour, and this will be in charge of carrying out the chemical analysis of the method used. The researchers, for their part, are charged $43 per hour, and they are responsible for distilling the joint chemical information to the research chemical staff. And finally, the chemical assistants will have a salary of $25 per hour, and will be in charge of assisting an analytical chemist in the investigative process and performing the sequential analysis. For the analysis by ion exchange chromatography, the use of nitrogen, phosphorus, and potassium was used. This ion exchange chromatography technique retains the analyte molecules on the column based on coulombic ion interaction. The ion exchange chromatography matrix consists positively and negatively charged ions. Essentially, the molecules undergo electrostatic interaction with the opposite charge in the stationary phase matrix. Thus, nitrogen, phosphorus, and potassium were used as standard solutions. This cost 42 for a 125 milliliter bottle for nitrogen, 224 for a 1000 milliliter bottle for phosphorus, and 19.7 for a 500 milliliter bottle for potassium. On the other hand, the atomic absorption spectrometer with graphite furnace technique was used to analyze magnesium, copper, and iron ions. This graphite or electrothermal furnace atomic absorption spectrometry technique has found the wide application for the determination of trace elements in water, soil, and biological samples. The detection limits are extended and under favorable conditions can reach picograms level corresponding to parts per billion concentrations. For this, it was used in the purchase of magnesium for $19 per bottle of 100 milliliter. For copper, what used on $100 for a 125 milliliter bottle, and for iron are used $42 per 100 milliliter bottle. Scientists with adequate experience are need to carry out this proposal. These are analytic chemists, researchers, and chemical assistants. The number of personnel experimentation is almost small because this analysis process takes a relative, relatively short time compared to other methods, and everything is implemented in the same experiment. The final total cost of labor will be depend on the duration of this research.
In the budget for the materials, various glassware was included for both techniques. Between these, volumetric flasks of 100 and 250 ml beakers, volumetric pipettes, and fluorescence flasks were included. The cost of this glassware is provided by wholesale, so the analyst will not necessarily use the entire amount purchased during an experiment. The prices of this each glassware are for 250 ml volumetric flask is about 2,800 for 30 of this one with each cap. For the volumetric pipettes, the price is 111 for the count of 10. For the volumetric flask of 100 ml, the price is 1,232 for the count of 30. And finally, the price of 1 liter fluorescence flask is about 624 with 50 cents for the count of 6. Also, to glassware, an ion exchange chromatograph was used to carry out the ion exchange study of the macronutrients, which are nitrogen, potassium, and phosphorus. This instrument has the cost of $38,999. In addition to this equipment, a Perkin armor atomic absorption spectrometer with a graphite furnace will be used to analyze the micronutrients as ferrum, copper, and magnesium. Before purchasing any of this equipment, it must be verified if they are available in the laboratory and that their operation is optimal, to then ensure better results of the practice. To be able to use the GFAAS spectrometer and save time in the analysis, it will be using integrated to this a graphite furnace autosampler. With this autosampler is included an autosampler vials, which replaces the insertion and supply process of the sample by doing it by itself. This instrument is also available in the laboratory, and the price for its vials is about 19 with 30 cents for a package of 100 count. Before beginning the ion chromatography analysis, is needed to filter all liquid samples with with a Buchner funnel of 150 millimeters of diameter connected to 1 or 2 feet rubber hose, which is also connected to a vacuum source. The funnel has an 0.45 micrometers filter paper inside. Filter After the filtration, the sample is transferred to the vials using a 1 ml syringe. The price of the syringe is about $197 for each one.
To summarize what has been said in the presentation before, here's the lean canvas proposal. We first start with the problem. The problem in this case is the high concentration of micronutrients such as potassium, nitrogen, and phosphorus can prejudice the plant growth and foliage. The low concentrations of micronutrients such as copper, iron, and manganese can also stifle plant growth. We are proposing a solution to determine the quality of the 2020 foliar fertilizer. We will be quantifying both micro and micronutrients concentrations in a sample. Our proposal is unique and innovative because there is no experiment that already involves the analysis of both micro and macronutrients in just one experimentation. Before you would have to do these two analyses apart, and then this means a higher cost and more time. So our procedure means there would a lower cost of materials and less time. We would also obtain the results in one single experiment instead of having to do different experiments. And then the people that can find this experiment useful are anyone who works in analyzing fertilizers, works in the agronomy field, or landscaping managers. And anyone who works with this can adapt this experiment, or they could hire, us to do the experiment for them. The total budget for personnel is $156.71 per hour. For the reagents is $424.89 for reagents, and for the materials and equipment is $44,094 for the equipment, but the GFAAS and GF autosampler were not included in this price because the price could not be determined. So further analysis has to be done to determine the prices, and the total budget for all the reagents, materials, and personnel per hour is $44,776.59.
The concentration of macronutrients and micronutrients in a sample were determined through the use of ion chromatography, graphite furnace atomic absorption spectroscopy, and flame atomic absorption spectroscopy. A calibration curve was constructed for each of the analytes, and this was used to determine their concentrations, 6.37 and 15.77. Pichea, voy a leerlo otra vez porque leí esos números fatales. Puedes borrarlo, espérate, 4. Ok, so, ¿empiezo la oración o empiezo todo? Ok. After dilutions, these concentrations... Sorry. After dilutions, these concentrations were determined to be 4.56, 0.59, 8.13, 36.37, 15.67, and 60.39 ppm for nitrogen, potassium, phosphate, manganese, copper, and iron respectively. The values for IC show high percent relative error, which could be due to the manual integration of signals. The percent of error for atomic spectroscopy were lower, as the data was already integrated. As for the linearity of the calibration curves, all exhibit large F values and R squared close to 1, which indicate linearity of the curves. Yeah. ¿Por qué? Ah,
These are the references. And if you have any questions, you can ask them in the designated forum. Thank you.